Ciao and welcome to the Salmon Scientist podcast. Today we're referring to our Brown, who is a PhD candidate at the University of Otago. She is studying the measles virus related to immunology, and in this podcast she shares what her research involved, what it's really like working in the lab, and how this work can connect and help our wider Pacific communities.
As well as like the molecular stuff we do, cloning, we use a lot of bacteria for that stuff, so we do it all here. As you can see by all the crazy protocols, optimizing, MIDI preps and mini preps all here as well. And then this is kind of like our molecular admin area. All our gels done here. And we go through a lot of pipette, so this is our refilling station. New minus 70 recently, nice. Very flash.
I love a freezer.
Yeah. We had an old one, it was minus 80, but they've changed it now that we've all got new freezers. They've said minus 70.
So Oa is a Samoan scientist researching measles and the biology behind it, how it affects our communities. Her work gives us a closer look at the science, the techniques, and the dedication it takes to turn research into impact.
And I kind of like it because it protects
the front.
Yeah.
Each lab here has a different coat colour and like the names.
That's cool.
This is all my stuff. It was kind of perfect. You came today. I was like, oh yeah, perfect. I don't have to be in the lab. I was like, actually, what am I going to do for her? What am I going to show her? But there are some dead cells in there that I can need to play with. Okay, this is going to be a bit loud. Did you do a lot of like cell work?
No, I did like a little bit on like a summer studentship.
Okay.
And like understanding that there's different types of containers, and they can sometimes like, what is it, like stick to the bottom if you're doing stuff like
flasks?
that. Yeah. Of the flasks. And they're very temperamental. Yes. And, everyone's like, my cells died. I was like, oh, that's just kind of normal.
Yeah, pretty much. So a lot of us have cells to look after. Down here.
Oh yeah, could you describe it? Why cells? Like, what are these?
Why cells?
What are cells?
So we've got, in this lab we work with PBMCs as well as cell lines.
Oh, okay.
So I don't know what the main difference is. So PBMCs are isolated from our blood, whereas cell lines are kind of immortalized from different things. So I've got HEC cells, which are human embryonic kidney cells, I think. So there's different cell lines for different purposes.
So like, instead of getting a live kidney a kidney from someone, it's easy to work on cells because we can use them in the lab and they're easy to look after. Like, is that
pretty much
kind of why?
Yeah. I was up here, and this is my baby flask. Do I say they're dead or? So TROK1 cells, they're adherent. They're good for transfection and expression, and they do not have the receptors for the measles virus, so that's why it's ideal. But yes, and I usually look at them under the microscope and put them in there, change the media. I don't know, should I play around with some stuff?
Yeah.
Just play with some PBS maybe. Oh, here we go. What is that? So a big part of looking after cells is passaging them. So every 2 to 3 days you check the confluency under the microscope and you split them because they continue to proliferate.
So we can't actually see the cells, is that right?
Yeah. Or usually when it's confluent you can kind of see like a blur because all the cells are adherent. Can't really see it there. Yeah, but usually when cells are confluent you can kind of see a sheet of cells at the back. But passaging for these specific cells include washing them with PBS, spinning them, counting them, and then resuspending in their media for them to keep growing at a healthy rate.
Behind every experiment is a bigger question. How do we understand measles well enough to better protect Pacific communities? The lab techniques shown here are part of the answer, helping scientists study cells, proteins, and immune responses in detail.
Oh, I'll keep this there. So,
So when you come into the lab, this will be part of your kind of like what you do in the lab during the day.
yeah, so pretty much if I've got cells, I'll look at them. It's supposed to be every day just to monitor whether or not they're growing well.
Can we see them?
Yeah,
like,
definitely have a look.
I wonder if I can see them down here. Or maybe not. I'm scared of the lens. Oh, we can.
Should I play with the focus?
Whoa. That's cool.
Yeah, so different cells will have different morphologies as well.
So the cells are like the little, how would you describe them?
They kind of look spindly, like almost eye-shaped. These cells, lots of adherent cells kind of have spindles. I guess it helps them to adhere to the bottom of the well, to the flask. But yeah, typically we come into the lab. If I haven't got anything set up, then I'll come in, check my cells, just like that. If they look confluent, then I'll split them how I showed you before. Otherwise, I will be transfecting at 8 a.m. or staining. I could take you down to the flow cytometry state.
That'd be
So
cool.
we do a lot of spectral flow. We can use that to kind of see the expression. It's quite good as quantifying as well.
Cool.
Compared to like Western blots and things like that, where
Yeah.
it's just like a yes or no, whereas you can kind of get numbers from flow cytometry, which is pretty cool.
I wonder if we do that. I've heard of it, but
Yeah.
I don't think I've done it. No.
It's a very immunology based technique.
Okay, yeah, I
like
wouldn't have done it.
Yeah, but I'm kind of trying to make it work for myself. I find it so much easier than a Western blot. I'll take staining over Western blot any
Oh my
day.
gosh,
Yeah.
Western, yeah.
No.
There's just so many steps. I mean, our lab, that's what they do every day. Like the students do.
Oh,
Western
really?
blots. Yeah. They'll have like boxes of, what is it, like the strips where they, soak overnight and stuff.
Yeah,
It's
yeah,
just
yeah.
like, yeah.
Constantly full.
So like, that experts at it, but it takes a long time
Yeah,
to
it
learn
does,
a Western
yeah.
blot. And to get a really nice image.
Yeah, a clean image
clean
of it. Yeah.
image.
I think that's what we were doing with our gels recently. We've got lots of gels, lots of empty things,
Oh my
lots of
gosh.
smears, molecular things, and the scientific things that you're doing, as well as the community outreach as well.
Because
So everyone over here, we do this stuff too.
Because you have to have those scientific skills to explain the science.
Exactly.
But then the community kind of look to you like, but how does
Now
that help
what?
us?
So what?
Exactly.
So this is our flow suite. We've got a Cytec Aurora and a cell sorted.
what
It's
do these
usually
do?
what Cytec do? You can look at an array. It's kind of like you just stain with antibodies. So whatever gene of interest or cells of interest, you can buy antibodies on certain colours or fluorophores. So you can look at heaps of colours. It's not really recommended, but you can get up to like 24 colour panels, and you can look at 24 different markers in one cell, which is really cool.
Wow.
But yeah, they use like lasers to kind of identify the populations in your sample.
And will it give you like a number, like a peak? What's
Yeah.
the
So it
outcome
kind of gives you have like a positive and negative, so you have single stain controls, and it tells you how much of your sample is expressing whatever
that's
gene of interest or cell of interest.
Ah.
Yeah.
And what do you put into the machine? Is it like
You could put a 6-well
Like a well
plate
plate.
or
Oh,
the
well.
fax tubes in there.
X tubes? I haven't seen them.
Yeah, but a lot of us, if we're doing lots of samples, we'll just put our 96-well plate straight on.
Okay.
That makes it easier too. Then cell sorting, I've never done cell sorting, but I'm pretty sure you can just put a whole bunch of samples in there, and then it kind of sorts your different cell populations
Oh.
if you want to work with specific
That sounds
cells.
really nice.
Yeah, it's super cool.
I can see, how you do that by hand normally.
Yeah,
but
yeah.
having a machine to do
Looking for the cells.
Oh my gosh.
So it'll sort your cells based on whatever markers you have, and then you've got these different populations of cells that you can use if you want to do different experiments with different populations, which is really cool. I haven't done that stuff, but
What machine do you use?
Da,
Oh, this
da,
one.
da.
And then, so you'll put your samples in,
Mm-hmm.
and then how long will it take to go through the analysis?
It'll depend how many samples I have. So maybe a tube or a well takes about 3-4 minutes.
Oh, quite quick.
Yeah, so it depends how many samples you have. But yeah, then they'll spit out some fax files, and then we just analyze on Flowjo. Is what we like to use. Just a bit of software that helps us sort through all our data.
And is it numbers that you're looking at when you analyze it?
Yeah,
Or is
you
it
kind of get like a proportion. You can get like percentage. This percent of your cells of interest are expressing this. You can also get like a median. Like they take in consideration all your data sets and kind of compares it across. A lot of like bacteria, more microbiology.
Oh, interesting.
Bacteria stuff, and then sixth floor, that is Peter Frenner and then Cook Lab, so they do TB and then CRISPR. Yeah, we're kind of separated by research interest, kind of.
Yeah,
between the floors.
makes
Yeah.
sense.
But yeah, this is one of the plate readers. There's 2 plate readers in the PCR room. Yeah.
And what do these do?
They can measure different kinds of things. I think there's a plate in there at the moment, but for me, some of my constructs have GFP or luciferase, which are just, they kind of just fluoresce different colors. So when you put your plate in the plate reader, they can just measure the intensity or the luminescence of that.
So it's like a key tool that you use in your day-to-day to really understand the science behind the measles.
Yeah. So after the flow cytometer, it spits out all the data, and then you just kind of sort through files. I can kind of walk you through this one, so you can gate different populations. So what's here? So I have my hex cells. So each different cells have like different size, so you can kind of gate side scatter, forward scatter to kind of find your cells. So my cell's kind of big, right there. And then you can also gate for single cells and doublets. So I'm looking at the single cells. We can stain for live dead to make sure what we're looking at are live cells. And then these are my 2 markers that I'm looking at the moment. So this antibody binds to a certain conformation of the ETH protein, and then the flag tag is just to see whether or not it's being expressed on the surface. And so you've got double positive here and double negative here, and we see that most flag positive are also in this certain conformation that I'm looking at, which is pretty cool.
What looks like routine lab work is actually part of a much bigger effort to create knowledge that matters. This research is more about science. It's about using science to support the health and well-being of Pacific peoples.